so I realized after class last time I did not start off with the limerick, so I better start off with the limerick or I'm going to be in trouble. So let me start off with the limerick. I confess with a bit of hostility. I succumbed due to some gullibility. My info got took by a mean online crook possessing a great lie ability. Okay, that was the limerick from last time. Here's the limerick from this time. In choosing a theme for the rowers, the admin gave what they asked for. A clever cliche he inverted to say, no ifs, ands, or buts, just ors. Not even a groan? Okay. Well, this will make you groan if that didn't make you groan. So, there's the real groan, right? So, all right. We'll have a little fun today. How's that? So, I have unfortunate news on the exams. The exams will not be ready to return to you until Monday. Yeah, I know. The TAs worked very hard. The TAs, in fact, have just finished them. I have not had the time to record the grades, and that has to happen before I can give them back to you. The exams will be available, I can tell you this much, on Monday morning starting at 10 a.m., not before then, so don't bug the office. The office tells me that people have been in there bugging them this morning, and that's not appropriate. Okay? So don't bug them before 10 a.m. on Monday. The BB office, they're going to split it up. They're going to have people in A's through Z, A's through G's in various places to pick up your exam there. So they're going to streamline it. It should be faster than it has been before. That'll start at 10 a.m. That will go until 11:30. And then they're gone over the lunch period, and they will start again at 1:30 and go until 4:30. So if you can't pick it up on Monday, then you will need to get it from your TA as we did last time, and the TAs will have it available for you in recitations. Okay? Yes? What if your recitation's at 9 on Monday? I never thought about that. That's a good question. I think most likely that I would just leave it at the 10 o'clock. And if you're in the 9 o'clock Monday recitation and for some reason you can't get it at 10 or whatever, I will make an exception for those, and you can talk to me about that. Okay? But I don't want to start things before 10. Okay? Okay. So that's coming. And again, they worked very hard, but they had an exam themselves on Monday, and just grading 400 exams is a lot of effort. All right. Well, last time I went through the pathway of gluconeogenesis, and I went through a little rapidly, so I want to slow down and talk a little bit more about some of the enzymes that are involved in gluconeogenesis. I will remind you of a couple of things. First of all, that 7 of the reactions in gluconeogenesis are simply the reversal of 7 reactions from glycolysis. You know that there are 10 reactions in glycolysis, and there are 11 reactions in gluconeogenesis, and that includes that first 2-step that I talked about last time that I'll mention briefly here again today. Okay? So we have the 2-step to bypass the Big Bang enzyme in glycolysis, and then we have reactions that bypass the PEPCK and the hexokinase reactions of glycolysis with different enzymes in gluconeogenesis. Okay. Gluconeogenesis, as I noted, occurs in the cytoplasm with the exception of the very first reaction and the very last reaction. The very first reaction is the one that you see on the screen, and it occurs in the mitochondrion of cells. Okay? This reaction takes pyruvate, which is a 3-carbon molecule, which is the end product of glycolysis, and uses energy from ATP to put a carbon dioxide onto pyruvate and make oxaloacetate. Oxaloacetate, as I noted before, is a 4-carbon molecule that you will hear a lot about next term because oxaloacetate appears in numerous metabolic pathways. This includes the citric acid cycle, which is the first thing that you'll learn about next term. It includes several amino acid metabolic pathways as well. So oxaloacetate is a very common intermediate that we see in pathways, and of course here we see it in gluconeogenesis as well. Okay, so that reaction is this reaction that you see on the screen is catalyzed by the enzyme known as pyruvate carboxylase. And I just wanted to say a brief word about pyruvate carboxylase. Of course, it's found in the mitochondrion. And the other thing about it is that pyruvate carboxylase uses a specific coenzyme. I've talked about coenzymes earlier in the term, where these are things that are non-amino acids that are part of a protein or part of an enzyme that help the enzyme to perform its function. The coenzyme that's associated or actually bound to pyruvate carboxylase is known as biotin. Biotin is one of the nutrients that we need to have in our diet. And biotin is a very common coenzyme for any reactions that involve carboxylation, that is putting a carboxyl group on. So you're seeing the addition of a carboxyl group here. You will see other places in metabolism where carboxyl groups get added to something, and biotin is almost always involved wherever there's a carboxylation. Biotin actually holds on to the carbon dioxide for the enzyme and carries it to the reaction so that that carbon dioxide can be put onto the molecule of interest. In this case, the molecule of interest is pyruvate. Okay, there's biotin, blah, blah. Don't really have to worry too much about that. Biotin is attached to a lysine side chain in the enzyme, and that's biotin down here where you see the blue and red circles down here. And this is what it looks like when it has bound to the carbon dioxide. So this is actually a carbon dioxide attached to biotin. There's the lysine side chain. Of course, you're not going to have to draw that or anything, but just to orient you and let you take a look at what biotin is actually doing. Okay, the second enzyme of gluconeogenesis is the second enzyme of the two-step, and that's this enzyme called Phosphoenolpyruvate carboxykinase. And as I noted last time, you're most likely to call it PEPCK. That's what most people call it. PEPCK is located in the cytoplasm of cells, and PEPCK is not found in very many cells. Okay? So you may remember that I said last time that gluconeogenesis does not occur in all cells of the body. It only occurs primarily in the liver and also in certain cells of the kidney. It's also in some of the gametes as well. Okay? This enzyme is one of the regulated enzymes of gluconeogenesis, and its regulation is primarily on the basis of whether or not the enzyme is actually made. Okay? So this is yet another mechanism for controlling enzymes. We talked about allosterism as a mechanism. We talked about covalent modification as a mechanism. And now here's a third mechanism, controlling an enzyme's activity by determining whether or not the enzyme is even made. If you don't make the enzyme, then of course you've completely controlled the action of that enzyme. And because of this control of PEPCK's synthesis in cells, that really determines where in the body the gluconeogenesis can occur. So skin cells, for example, don't make PEPCK, and they therefore can't make glucose in the process of gluconeogenesis. The reaction catalyzed by PEPCK is the one that you see on the screen. And you'll notice that there's another triphosphate that is used. And in this case, we're converting a 4-carbon intermediate back to a 3-carbon intermediate, which means there's a loss of carbon dioxide. And this loss, which requires the triphosphate, in this case, requires GTP. And as I noted last time, GTP has the same energy as ATP. So these two reactions are taking a total of 2 triphosphates to go from pyruvate back to PEP. And that's not inconsistent with what I told you with how much energy there was in the PEP bond. Remember I said there was almost enough energy in there to make 2 ATPs. You can see that going backwards, it takes 2 triphosphates to go backwards. So not too much of a surprise with respect to that. Okay, so that's PEPCK. Yep, come on. There we go. Yes, question? Yeah, Deidre. Oxaloacetate, Yes. Right. So the question was, first reaction I talked about, the pyruvate carboxylase in the mitochondrion. This reaction that I just showed you, the PEPCK reaction, is occurring in the cytoplasm. And cells have to therefore have a way of getting oxaloacetate from the mitochondrion out into the cytoplasm so it can be used. You will learn next term of a shuttle that cells use to move oxaloacetate out to the cytoplasm. In fact, I'll briefly tell you about it here because it relates to the topic of obesity that we talked about before. Okay? So one of the things that you'll discover next term when you talk about the citric acid cycle is that the citric acid cycle is totally dependent on NAD. Okay? Citric acid cycle has 3 places in it where it uses NAD. When the cell is low on NAD, we've already talked about how fermentation is what the cell goes through to do that. And they might think, well, when will the cell be low on NAD? One is when it doesn't have oxygen, okay? But the other is when the electron transport system isn't running. Okay? When the electron transport system isn't running, there's 2 ways that could happen. One is if oxygen is missing. The other is if you're not burning anything up. You're not exercising. So if you're eating a lot, you're sitting around, and you're not burning up things, then the electron transport system isn't running, which means that NAD concentration will be low. Okay? NAD concentration will be low. So when the NAD concentration is low, the citric acid cycle stops. And when the citric acid cycle stops, the first intermediate of the cycle increases, and the first intermediate is known as citrate, which is what gives the cycle its name. And citrate is made from oxaloacetate and acetyl-CoA. Okay? So when the citrate starts to accumulate in the mitochondrion, which is what's going to happen when you're not burning off as much as you're eating, when the citrate starts accumulating in the mitochondrion, and by the way, I'm just telling you this, you don't need to know this. So this is just for your own interest, okay? Don't throw a big curveball with you at the citric acid cycle at this point. So you can relax about that. As the citrate starts to accumulate, the citrate actually is the molecule that cells use to move oxaloacetate out into the mitochondrion, out into the cytoplasm. Okay? So citrate moves out, and when it gets out, the citrate is broken down into oxaloacetate and acetyl-CoA. Okay? Fine and dandy, except for the cytoplasm is also the location where fatty acids are made. And the precursor of fatty acids is acetyl-CoA. So when you're not exercising, you're slowing down or stopping the citric acid cycle, and you're moving these things out into the cytoplasm that's going to favor the production of fat, or ultimately through fatty acids. Okay? So a long answer to a short question. Okay? All right. Now we'll go back to the things that you are responsible for. Okay. The other enzyme that is different in gluconeogenesis, that's not shown on my screen here, but that is not found in glycolysis, is the enzyme known as fructose 1,6-bisphosphatase. Or as I said, you could call it F16-BPase. This enzyme, I'll say a little bit more about later, but this enzyme does not catalyze the reversal of the PEPCK reaction. Instead, what it does is it takes fructose 1,6-bisphosphate and clips off the phosphate on position 1. That's an energetically favorable reaction because you're removing a phosphate, and it makes what would otherwise be an energetically unfavorable reaction be an energetically favorable one because it's not reversing the pathway of making ATP, which is what it would have to do if it were reversing the PEPCK reaction. Question? Oh, yes. Phosphate on position 1. Okay? So you have fructose 1,6-bisphosphate becomes fructose 6-phosphate. Okay? Similar mechanism used in this reaction for this enzyme here, known as glucose 6-phosphatase, which is doing the backwards of the hexokinase reaction, but again, it's not reversing the reaction, it's simply clipping a phosphate off. You can see that happening right here inside the endoplasmic reticulum. So here is the enzyme in the membrane of the endoplasmic reticulum. We see that glucose 6-phosphate gets moved into the endoplasmic reticulum, and then there's a hydrolysis reaction, that's why water is involved, to clip off that phosphate and yield free glucose. And then free glucose is dumped back out into the cytoplasm, and ultimately once in the cytoplasm, of course, it can be dumped out of the cytoplasm into the extracellular space, which is what the liver is doing, because the liver is going to take that glucose. The liver doesn't need the glucose. It's got plenty of glucose, and it's going to put it back out into the bloodstream so the body can use that glucose. The liver plays, as you will see, a very, very critical role in balancing several things in the body. It balances the level of glucose, which is what we'll talk a little bit about here today when we talk about the Cori cycle. It balances the level or the production of fats and fatty acids, and it also balances the level of cholesterol. So the liver's playing some really, really important functions in terms of balance, metabolic balance inside of our bodies. Okay, questions on that before I talk about regulation? Yes? So a question about biotin, is that involved in the carboxylation of prothrombin? I believe the enzyme that does that uses biotin, yes, but biotin is not an enzyme. Biotin is a coenzyme. That's something that's attached to an enzyme that helps that to happen. So that carboxylation reaction that puts the carboxyl group onto the side chains of glutamic acid in prothrombin is, in fact, pretty sure using biotin as a coenzyme. Okay? Yes? Yes? Okay, so the question is, I talk about glucose being a poison, and here's the liver dumping a poison out into the bloodstream, right? So this is a poison in a figurative sense. That is, high levels of this compound, glucose, are going to cause a problem for cells in the body. You can rest assured that your liver is not putting out high levels or poisonous levels of glucose to cause problems for you, all right? So it's a good question, but that's not a problem in terms of what's there with the liver, okay? One of the reasons that people look at the Atkins diet and they say that the Atkins diet makes some sense is because the glucose levels in the bloodstream don't go high-low because people are getting carbs. And in fact, the glucose levels in the bloodstream stay at a much more manageable level because the liver is really regulating that. And the liver will regulate that at a very good level if there aren't other factors to consider. But yeah, okay? Okay. Well, let's talk about regulation, because I've already mentioned regulation for glycolysis. Now we're going to overlay the regulation of gluconeogenesis onto the glycolysis scheme. This introduces a concept that we need to consider, and you'll see this concept arising when we talk about glycogen metabolism as well. And the concept is that of reciprocal regulation. Reciprocal regulation, okay? Well, as the name suggests, reciprocal means opposite, and regulation means regulation, right? So reciprocal regulation occurs when the same compound or the same action has opposite effects on catabolic and anabolic pathways. I'll repeat that. That's an important definition. Reciprocal regulation occurs when a molecule or an action results in opposite effects on anabolic and catabolic pathways. We're going to see several examples of it here in these two pathways of glycolysis and gluconeogenesis. Remember, of course, that glycolysis is a catabolic pathway. Catabolic pathways involve breakdown of bigger molecules into smaller ones. They involve oxidation, and they involve release of energy. Okay? Gluconeogenesis is an anabolic pathway. Anabolic pathways involve the synthesis of bigger molecules from smaller ones. They generally involve reduction, and they generally involve input of energy. I didn't mention it as I showed you the pathway for gluconeogenesis, but the song that I sang the other day actually mentioned it, and that is to start from pyruvate. If you count the number of ATPs it takes to make a glucose, starting with pyruvate, the answer is it takes 6 triphosphates. Okay? 6 triphosphates to make a single glucose from 2 pyruvates. Right? So keep in mind that we only get 2 ATPs if we break down glucose. So this sets up a dynamic for cells that cells want to avoid. Right? What does that mean? Well, let's imagine I'm a cell, and I have glycolysis and I have gluconeogenesis going on simultaneously. Okay? That's very possible because most of the reactions are going on in the same place in the cell, right? Imagine they're both going on simultaneously. What's going to happen? Glycolysis is going to go through, starting with glucose. It's going to make 2 pyruvates. It's going to make 2 NADHs. And it's going to make 2 ATPs, right? Those are the products of glycolysis. Gluconeogenesis says, oh, here are 2 pyruvates. I'm going to grab these 2 pyruvates, and I'm going to make glucose. Well, to grab these 2 pyruvates and make glucose, it takes 2 NADHs, so there's a balance there. But it's going to take 6 triphosphates to make glucose. That's a net loss of 4. So if I were to have both pathways going on at the same time, I would start with glucose, I would go down, I would end up with glucose, and I would have lost 4 triphosphates. And I'd have nothing to show for it. Nothing to show for it. So what I've just described to you is something that we refer to as a futile cycle, F-U-T-I-L-E. A futile cycle occurs when catabolic and anabolic pathways are occurring simultaneously. Okay? So it's not in the cell's interest, usually. There are occasionally places where it is in the interest of the cell, and I'll mention them briefly. But it's usually not in the cell's interest to have both catabolic and anabolic pathways going on at the same time because all the cell is doing is wasting energy, and it's getting nothing for it. Because of this, cells have protections built in that stop this from happening. And one of the protections that's built into this is reciprocal regulation. Okay? So how does reciprocal regulation work? Well, part of it works as you see on the screen. Let me show you a really good example. Let's imagine that I am, don't look at the screen for the moment, but let's just imagine that I am a cell and I am burning up a lot of my energy, and my ATP levels start to fall. Okay? I'm going to need some energy. I'm going to need some ATP, right? My ATP levels start to fall. When my ATP levels start to fall, my ADP levels start to rise. And if I don't have something converting ADP back to ATP, then I start using ADP for energy, and my AMP levels rise. So AMP in a cell is a really good indicator of the fact that this cell is low on energy. If you look at the phosphofructokinase reaction, you can see that one of the things that's in green, meaning go, is AMP. AMP activates phosphofructokinase. That makes sense. The cell is low in energy. When the cell is low in energy, it needs to make ATP. The low in energy indicator is AMP, so phosphofructokinase gets activated, and glycolysis proceeds. Glycolysis is going to lead, ultimately, to the production of ATP. If we think about this cell being at a very low energy state, at the point where we have AMP concentrations high, we think this would not be a real good time for the cell to be making glucose. Right? Because it's going to take energy to make glucose. I don't have much energy sitting around, so I don't want gluconeogenesis going on. And when I look over at the fructose 1,6-bisphosphatase, look what turns it off. AMP. Okay? So low energy is turning on glycolysis, which produces ATP, and turning off gluconeogenesis, which is using ATP. Reciprocal regulation. Okay? If you look at the fructose 2,6-bisphosphate, you see that it has exactly the same effect that AMP has. It activates phosphofructokinase, and it stops the activity of fructose 1,6-bisphosphatase. Again, turning on glycolysis, turning off gluconeogenesis. When we take those compounds away, then we start favoring fructose 1,6-bisphosphatase, and we lessen the effects, or we lessen the positive effects of phosphofructokinase. If you think about this, remember we already saw that when ATP levels start to rise, we turn off phosphofructokinase. ATP is an indicator of high energy. High energy, we don't want to be running glycolysis. It's turning off the enzyme very nicely. Here's citrate. Remember I told you briefly that citrate was being kicked out of the mitochondrion when you're not burning things up. When you're not burning things up, it means your energy level is high. Citrate is turning off PEPCK. It's favoring, okay, the fructose 1,6-bisphosphatase. So again, we're seeing reciprocal regulation. Now, you'll see as we look through here that not everything is reciprocal, okay? But many things are reciprocal regulators of pathways. The other things that we look at down here, and by the way, these things over here are not overly important in the overall scheme of things. I don't expect you're going to memorize the regulators of these things down here. These up here I think are worth knowing. These down here less important. But these are nonetheless important for us to consider or talk about. Okay? ATP is turning off pyruvate kinase. That's important. When are we going to be making glucose, folks? High ATP or low ATP? We're going to be making glucose when we have high ATP, right? Because we don't want to be making when we have low because we're going to waste ATP energy if we try to do it when it's low. So if ATP levels are high, we don't want glycolysis going on, so we stop pyruvate kinase in its tracks. And when we stop pyruvate kinase, the other reactions over here can catch up and start making glucose. Okay? You'll see the things that turn off these enzymes that make glucose are indicators of low energy. There's ADP and there's ADP again. You also see our friend acetyl-CoA that was coming from the mitochondrion when we weren't burning things up that's saying, hey, we've got plenty of energy. Let's make things into glucose. Okay. So this reciprocal regulation is very simple. It's like feedback inhibition. Remember, feedback inhibition was simple because it inhibited the first enzyme in the pathway, and we didn't have to worry about any of the other enzymes. This guy is using one molecule to really shut everything off on one side at the same time as it's turning everything on the other side. Pretty cool setup. Questions about that? You guys are ready to get to Turkey Day, aren't you? Question here. What's so bad about having high ATP? Well, I don't like to think about these things in terms of bad or good. But let's put it this way. When you have ATP sitting around, your cells are little bankers. Okay? They're bankers. Bankers take money, right? And they do something with that money, right? And the thing that they do with that money is they make more money. That's what bankers do. Cells are bankers in that they have the currency that the cells have is that of ATP. So cells are taking ATP, and it turns out that ATP is very transient. Okay? Very transient. Cells, ATP is not a good way to store energy. A, it's chemically unstable. Okay? Relatively unstable. All right? And B, it's obviously used for a lot of things. So when cells have an excess of ATP, they put it in their bank account and convert it into something like stocks or bonds that it can use. And the currency that the ATP is being converted into is fatty acids for the most part. Okay? So that's the bad part of what's there. Okay? So if you're not burning things off, then that ATP is going to be used to store things. And it, biologically, storing energy makes a lot of sense. Okay? Human beings evolved under conditions where we had fasting, we had famines, and then we had great abundance. And we went up and we went down. And being able to store energy when you had a lot to eat made very good and makes very good sense. So that when those times come when you don't have that energy, you don't have that food available, then you can live off of stores. Okay? Make sense? Okay. All right. I need to talk about a topic that is probably the least favorite figure that I will show the entire term. Year in, year out, this is the one figure I'm getting ready to show you that students really don't like. Okay? I'm going to try to keep it simple for you and make it simple for you. And it's really not as complicated as I think you're going to make it when you first look at it. So what I would say to you is, when you look at this figure, don't take your initial reaction as your final reaction. Let it kind of just settle with you, okay? This figure involves the regulation of the synthesis of fructose 2,6-bisphosphate. I told you that fructose 2,6-bisphosphate is a very important molecule in controlling glycolysis and gluconeogenesis. Fructose 2,6-bisphosphate activates PEPCK, and it also inhibits fructose 1,6-bisphosphatase. Okay? By the way, just a sort of a reminder here, and I think you guys know this, but there are names of things that sound very similar. Fructose 1,6-bisphosphatase sounds like fructose 1,6-bisphosphate, right? The difference being the A's. And A's will always be associated with an enzyme. Don't confuse enzymes with molecules. So always pay attention to that ending in terms of what's on there, because that's going to become important when I talk about fructose 2,6-bisphosphate and fructose 2,6-bisphosphatase and related things. Okay, here's the figure you're going to love. Okay? Now, what this figure is showing is really the dynamics by which fructose 2,6-bisphosphate is made and broken down. Okay? The dynamics by which it's made and broken down. So how is it made? All right? Let's start with the synthesis of fructose 2,6-bisphosphate. Remember, this is not glycolysis. This is a molecule that regulates glycolysis. So you're not seeing reactions of glycolysis, even though you're seeing intermediates of glycolysis. And a common concern students have is, but if there's fructose 6-phosphate being used here, what about the fructose 6-phosphate in glycolysis? Which I remind students that there are billions of molecules of fructose 6-phosphate in your cells. Okay? So it's not like we only have one, which is the way we think about them in metabolic pathways. All right. Now, that said, let's follow the synthesis and breakdown of fructose 2,6-bisphosphate. Why do we care about it? Remember that fructose 2,6-bisphosphate stimulates PEPCK. So that's one of the reasons it's important. Okay? It inhibits fructose 1,6-bisphosphatase. The way it's synthesized is we start with a molecule of fructose 6-phosphate. We take some ATP energy, and we put a phosphate onto it, just like we did in the PEPCK reaction. In the PEPCK reaction, you may recall, we took fructose 6-phosphate. PEPCK catalyzed the addition of a phosphate to make fructose 1,6-bisphosphate, right? And I said at the time, we call that PEPCK, but more likely you're going to call it PEPCK1 because there is a PEPCK2. Now we're going to see the PEPCK2. PEPCK2 is right here. PEPCK2 catalyzes the addition of that phosphate, and in fact the name is very simple because PEPCK2 makes fructose 2,6-bisphosphate. PEPCK1 makes fructose 1,6-bisphosphate. So you've got an automatic memory thing there based on the name of the enzyme. PEPCK2 makes fructose 2,6-bisphosphate. End of story. That's all it takes to synthesize fructose 2,6-bisphosphate. It takes nothing more. Okay? And it's simply an enzyme, and it's an enzyme that has a catalytic action just like PEPCK1. Now, what this enzyme has, though, is something that's really interesting and cool. This is another kind of odd regulation that you haven't seen before. Okay? I told you that cells are control freaks. I told you that cells, when they have a way of turning a switch on, they've got to have a way of turning a switch off because they don't want to leave a switch on all the time because that's going to cause problems. If I left this switch on all the time, the cell would continually be making fructose 2,6-bisphosphate, which would continually be stimulating glycolysis, and it might mean that I'm running glycolysis when I don't want to if I can't turn that switch off. So to turn that switch off, I have to have an enzyme that's equivalent to the kinase, that is the PEPCK2. PEPCK stands for phosphofructokinase. And the equivalent enzyme for a kinase that does the opposite thing of a kinase is a phosphatase. In gluconeogenesis, you saw an enzyme known as fructose 1,6-bisphosphatase. Here are the phosphatase. And the phosphatase is taking a phosphate off of fructose 1,6-bisphosphate. To take a phosphate off of fructose 2,6-bisphosphate, we have to have a phosphatase. And the cell has a phosphatase. It's known as fructose bisphosphatase 2, or FBPase 2. You see it right there. It catalyzes the breakdown of fructose 2,6-bisphosphate into fructose 6-phosphate. We're back where we started, folks. We're back where we started. PFK2 makes fructose 2,6-bisphosphate. FBPase 2 breaks down fructose 2,6-bisphosphate. All right? Very simple. We make it, break it down. Now, here's the cool hook that I told you about. We don't see this hardly anywhere else in biology. I'm not aware of any other system that's like this. Okay? The system that's here is fructose 2,6-bisphosphatase. Fructose bisphosphatase 2 and PFK2 are on the same protein. You've got two catalytic activities that do exactly the opposite thing, and they're on the same protein. Talk about your futile cycle. Would you want to have an enzyme that both makes and breaks down a molecule on the same protein? Well, if you had that, it means you better have something very, very careful to regulate it so that they're not both active at the same time, or you're going to have futile cycle deluxe. Because that's right there. As soon as it's made, it's broken down, it's broken down, it's made, it's broken down, it goes back and forth. You got problems. You only want one of those active at any given time. Well, it turns out the cell has a way of doing that. Okay? If you look at this protein over here on the left, you'll see that it has no phosphate on it. And if you look at the protein over on the right, you see that it has a phosphate. Putting a phosphate onto this protein complex causes the phosphatase to be active, and it causes the PFK2 to be inactive. Taking the phosphate off causes the FBPase2 to be inactive and the kinase to become active. It's completely reciprocal. Only one can be active at a time. And then the question is, well, how does the phosphate get on and off? And this is something you've already seen. Protein kinase A. What stimulates protein kinase A? What's that? Cyclic AMP stimulates protein kinase A. What stimulates cyclic AMP? What hormone stimulates it? Adrenaline epinephrine, right? Adrenaline epinephrine is released when we need energy, when we need glucose, okay? And when we need glucose, what pathway is being stimulated? Gluconeogenesis. Our liver is being stimulated to dump glucose out into our bloodstream when glucose is low. It's one of the ways adrenaline's working. How do we take the phosphate off? We take it off with a phosphatase, and this phosphatase is called phosphoproteinphosphatase that takes the phosphate off of that protein complex. And how does phosphoproteinphosphatase get activated? By insulin. Remember that glucose is a poison. Insulin is there to deal with that poison. Insulin is causing the cell to take up glucose, right? If free glucose accumulates in the cell is hosed, so the cell must do something with that glucose. And one of the things it does is it breaks it down in glycolysis. This reciprocal regulation works all the way down to the level of the control of this interesting protein complex. Questions about that? Now, as I said, your reaction to this is going to be, oh, there's a lot of stuff here. Do it like and in fact, what I really recommend you do is not stare at it. Okay? Staring at something, I stare at that for hours. I'm like, well, you know, it's not Gilligan's Island, okay? You're not going to sit and staring at it is going to get you anywhere, right? But writing it down. Learn to write this out without looking at it. And as you do that, you will see there's not as much complexity to it as there is if you're staring at it. Don't stare at it, okay? If you have questions about how this works, then let me know. And I'll be happy to explain it to you in more depth. Okay? No comments, questions? Okay. Well, the last thing I'm going to talk about today is something that's very easy to understand. And this is the first thing we're going to talk about where you can really start to understand how your body works. It's a very simple thing, but it's a very interesting thing as well. It's called the Cori cycle. I'm going to describe it to you before I show you the schematic. All right? Let's imagine, if you will, I'm a muscle cell. I'm exercising. I'm exercising very heavily. And when I exercise very heavily, I'm going to use my ATP. As I use my ATP, I need glucose and I need oxygen. Because when I have glucose and oxygen, my maximum ATP production will be realized. 38 ATPs. Okay? As I start running out of oxygen, which will happen as I'm exercising very heavily, my ATP concentrations will start to fall, my oxygen concentrations will start to fall, and I'm going to have to start to ferment if I want to keep exercising. I don't want to die as a muscle cell that runs out of energy. So fermentation starts to happen. In fermentation, what's happening? Pyruvate is being converted into lactate, okay? And NAD is being produced to keep glycolysis going, right? So we're keeping glycolysis going by the virtue of the fact that we have the fermentation going on. Well, lactate starts to accumulate in the cell, and you'll notice I didn't tell you lactate went to anything else, because lactate doesn't go to anything else. The only thing lactate can go to is pyruvate. But what does it take for lactate to go to pyruvate? NAD. And the cell's already short on that. So lactate in an exercising muscle cell is of no use to the exercising muscle cell. Worse, lactate is an acid, and the cell is starting to accumulate protons. Remember the hemoglobin, the Bohr effect? I told you rapidly metabolizing cells were producing protons. This is a rapidly metabolizing cell. Lactate is a source of those protons. The muscle cell says, I don't want this lactate. So the muscle cell dumps lactate out into the bloodstream. Because it's of no use to it, it's also getting rid of those protons in the process of doing that. The lactate floating around in the bloodstream goes back through the lungs, through the heart, and it hits the liver. Okay? The liver is important. The liver is not an exercising muscle cell. The liver is very close to the heart and the lungs, and consequently the liver is full of oxygen. If the liver is full of oxygen, the liver is full of NAD. And it can take lactate and convert it into pyruvate very readily, because NAD is needed to do that. And the liver happens to be the place where gluconeogenesis occurs. So now pyruvate gets made into glucose because the liver's got plenty of energy. And glucose gets dumped into the bloodstream and travels right back to the muscle cells. I've just described the Cori cycle to you. Here's how it looks. Okay? In the muscle cell, upside down here. In the muscle cell, glucose is being converted into lactate because there's low oxygen. Lactate is dumped into the bloodstream. It travels to the liver where it goes to pyruvate, gets back to glucose, and this cycle goes on. This is a very cool cycle. So that even though your muscle cells could easily exhaust their own supply of glucose. The liver keeps them going. Okay? The liver is keeping the muscle cell going because it's continually dumping out that glucose so that the liver cell can take it up. And that's necessary because, remember, this liver cell is fermenting, and it needs 19 times as much glucose as if it's not fermenting. The liver is a great source of glucose for that muscle cell. Comments, questions? Yes? I'm sorry? Oh, yeah, I'm sorry. These are triphosphates. Yeah, thank you. I had to think about it myself. So these are 6 triphosphates. That's a dumb, hadn't noticed that before. That's a dumb thing there. So you're producing 2 triphosphates over here. You're taking 6 over here. But remember, the liver is full of oxygen, so it's got plenty of energy to do that. It doesn't have the problem of energy. The muscle cell is the thing that's lacking the energy. Garrett?
What happens
when liver runs out of If the liver runs out of oxygen, you're pretty much hosed, okay? Because the liver is right there, right next to the oxygen source. I'm sorry? What percentage of a decrease would it take? Is that the question?
Yeah,
I don't know. I don't know what percentage it would decrease. I don't know. Yes? Okay, so if lactate's being delivered to the liver, what causes lactate buildup in the muscles? Lactate buildup in the muscles is a little controversial in terms of whether it causes problems or not. But you can imagine that the bloodstream is not a specific thing. You see it here. Here's a blood vessel, takes it there, and it's as if it's not in one place or the other. But remember that these are capillaries around where all this is happening. And so it's not as clear-cut that it's not there. Okay? You guys want a song for the day? Okay, since you've just taken an exam, I have a song relevant to that. It's to the tune of an old Beatles song called Norwegian Wood. It's called Student Nightmares. I answered 3B, but then I thought it might be C. Or was the false true? I can't undo. It makes me blue. It asked me to list all the enzymes that regulate fat. As I wrote them down, I discovered I didn't know jack. I ought to give thanks, scoring some points, filling in blanks. I squirmed in my seat, feeling the heat, shuffling my feet. Professor then told me there wasn't a chance I would pass. So I started crying and fell through a big pane of glass. I suffered no harm, 'cause I awoke to my alarm. Oh, nothing compares to deadly scares of student nightmares. You guys know those kinds of nightmares, right? I still have them.